speaker for the session, Dr. Gina Gallego, who received her PhD from Washington State University in immunology and infectious disease, and is currently doing a postdoc within the Morgridge Institute for Research and the University of Wisconsin-Madison in the Department of Microbiology and Immunology.
Hello. Hello, everybody. Can you hear me well? Good. So I'm happy to be here to share with you the research vision of my future lab. That is understanding the metabolic changes induced in host cell by enteric apicomplexa parasites. But first, I need to introduce you what are these apicomplexa parasites. So these parasites are characterized by the presence of three organelles. They are rhoptia, micronems, and spherical bodies that sometimes they are called dense granules. Between this important phylum of parasite, we have different microorganisms. We have Toxoplasma, we have Cryptosporidium, Plasmodium that cause malaria, we have Theileria and Babesia that affect mainly animals, and all of them have very important, produce a lot of important morbidity and mortality in humans and animals. So normally in a parasitology talk, people present the life cycle of the parasites. But today, I'm going to present my research life cycle. So I started this research journey early back in Colombia, doing my undergraduate studies. I did my bachelor degree in La Universidad del Tolima, and I did internship. I worked for the immunology institution in Colombia, working in Plasmodium. I learned or I participate in projects in vaccine development. And then I also learned a lot of techniques in immunochemistry. Then I got my scholarship to move here to the United States. I did my PhD in Washington State University, where I studied Babesia. I learned a lot of molecular biology techniques and proteomics. And then I moved to Wisconsin. And in Wisconsin, I started working with Dr. Laurel Knoll, who is a famous parasitologist who studied Toxoplasma. But also, I get a multidisciplinary fellowship that have 2 PIs. So my second PI is Dr. Melissa Scala, who is an engineering and work more in cancer and developing tools to study cancer. So I'm the perfect mixture of both of them. So I work in parasitology and applying these engineering techniques. And I'm mainly in my postdoctoral studies studying Toxoplasma and Cryptosporidium, but in my future lab, I want to focus more on Cryptosporidium. So why these parasites are too important? So all of these parasites produce a lot of outbreaks around the world. Here I have some examples. So in Wisconsin, where I am located, in 1993 was a very important outbreak, maybe some of you remember, where people have to boil water even to brush their teeth. So in that outbreak, more than 400,000 people was affected. And here we have other example why these parasites are important. So as we can see, affect different parts of the world, are mainly transmitted by water, but also by food, affect mainly immunocompromised patients and children under 5 years old. Also here, other example, toxoplasmosis also produce a lot of outbreaks. Here, as we can see in this map, and also is transmitted by meat, vegetable, water, and milk. So why is important to study these metabolic changes inducing the host cell by these parasites? So it's very important because the parasite is a parasite, so it depends on the host to be able to replicate and produce a lot of metabolites. For example, the parasite cannot produce the purine rings, so they need to steal from the host. Also, it's important because the metabolism of the parasites behaves sometimes similar to cancer cells. Also, it's important because many of these metabolites could be very important drug targets. So now, this is my research vision of my lab, and I want to study, as I mentioned at the beginning, Toxoplasma and also Cryptosporidium. These are dysenteric apicomplexa parasites. And what really interests me is that there is a lot of data, or some preliminary data, that said that the infection with these parasites are a risk factor to develop colon cancer. So I want to try to understand how from chronic disease we can move to cancer, but I need to take little steps. So I have 3 aims that is going to allow me to know better the metabolism of these infected cells, and I will describe each of these aims. So I want to study mainly redox biology. In the second aim, I want to study more host nucleotidases. And in the third aim, I want to study more glycolytic demands. So let me start with the first one. That is why to study, why to understand the redox biology in the infected host cells. So this is important because there are many unknown mechanisms in redox biology. This redox biology influences immune response, and again, is a very important target for drugs development. So I use optical metabolic imaging technique, that is an engineering technique, to study these parasites. So using this technique, we are able to measure NADH and FAD in a non-invasive way in real or physiological conditions. So we are able also to calculate optical redox ratio and all these parameters that we can see here in this table. So using optical metabolic imaging in Toxoplasma gondii infection, in the same field of cell, we can have measure of NADPH, FAD. I use a cherry parasite to be able to distinguish between uninfected and infected cells. So using this approach, we found very important changes at the level of NADPH during 48 hours post-infection. These are infected cells, and in color you can see the fluctuations in NADPH. Also, we found fluctuation in FAD. So using other techniques, sometimes it's not very easy to have these measures. So I'm very happy using the optical metabolic imaging for that reason. Also, we found that the infected cells over time trying to become more oxidized. So here we see the same result by using quantitative way. We compare with the uninfected cell, and we see that the infected cell have a more oxidized redox ratio. So, but there are still many questions to answer. So in my future lab, I want to study Toxoplasma gondii infection in different organoids. We have in the lab growing placenta organoids, regna organoids, intestinal organoids. In Cryptosporidium, I want to study also in different colon cancer cells. And we want to study how the different stage of the parasite change the redox biology of these infected host cells. Also to test antiparasitic compounds and to check how they change the redox biology of the cells. Study the gene expression. For example, in Toxoplasma, there are at least 57 genes related to redox biology, and all of them, nobody have studied them. Also, I want to study the mitochondrial function and the oxidative stress. So now moving to my second aim, that is to determine the role of host nucleotidases in enteric apicomplexar infections. So I want to study that because, as I mentioned at the beginning, parasites cannot produce their own purines. So this is a required, to study host nucleotidases is required for parasite replication of the host nucleotidases are important for parasite replication. Also are target drugs, and there are many unknown mechanisms in this pathway. So I study, so this is a normal picture of the invasion process on Toxoplasma. So as we can see here, Toxoplasma kills the host cell and secrete a bunch of proteins, so I call this kiss and spit. And then create this parasitophos back wall where the parasite grow and replicate inside the host cell. So at this point I was wondering if maybe this single kiss can change the metabolism of the host cell. So in order to study that, I use an inhibitor of actin inhibitor called cytocalaxin D that allow the kissing and spit but prevent invasion. So using this technique, I did a metabolomics analysis to see if the kiss and spit change the metabolomics of the host cell. So I studied this during 12 hours post-infection, and I found significant difference at nucleotide levels, glycolysis level, pentose phosphate pathway levels, and amino acid level. So I confirmed this result with glucose tracking. And also here I want to compare the kiss and speed metabolomics with our previous published full infection metabolomics. And we found in common these 9 metabolites. Now, here I want to highlight 2,3-bisphosphoglycerate and these purines. So 2,3-bisphosphoglycerate is an allosteric activator of one enzyme called cytosolic nucleotidase 2. And this enzyme participates in the production of purines. So let me explain this hypothesis. So I believe that the kiss and speed of Toxoplasma gondii increase the abundance of 2,3-bisphosphoglycerate. And this one activates the cytosolic nucleotidase 2 enzyme that phosphorylate GMP to produce guanosine. And this guanosine is taken by the parasite to be able to create DNA and replicate. The same happens for IMP. IMP is phosphorylated by CN2 enzyme in order to produce inosine, and inosine is taken by the parasite. In order to test this hypothesis, I use a drug called fluorodavine that inhibits CN2. I test at 3 different concentrations, and the drug inhibits the replication of the parasite over time. So then I run metabolomics in these 4 different conditions in the presence of fluorodavine and without fluorodavine. And what we found was, I will focus here in the purple condition. We found that the products, GMP and IMP, was increased, and the products, the substrates, GMP and IMP, were high in abundance, and the guanosine and the inosine, that are the products of this reaction, were downregulated. So it looks like the CN2 also is affecting, we confirmed that the CN2 is affecting the production of these purines. So also other technique that I use is click chemistry to evaluate the inosine incorporation. So we evaluate the inosine incorporation in 4 different conditions, and we found there is more inosine incorporation in the presence of the CN2 knockout hot cell line. So there are many questions to answer because this is a very complex pathway, how the parasite take purine from the host cells. So I want to continue this study using click chemistry, nucleotide metabolic profile, and intestinal metabolic cartography of a spatial cartography, and gene expression and knockout of CN2 in other different cell lines. Finally, my aim 3, that is to study how Cryptosporidium parvus manipulate the host to meet parasitic glycolytic demands. Again, it's a known mechanisms, are target for drugs development, and is key to understand this colon cancer development. So I did some preliminary experiments infecting mites and treating with STAC, that is a protein licensed from Toxoplasma gondii, and STAC reduced the infection of Cryptosporidium. So then I moved to do a gene expression of the cocoon and ileum of these infected and uninfected mice, and we found that this solution stack activate the interferon type 2 response. But in addition to that, what I want to highlight here is that the stack also produce downregulation of 3 important metabolites, these 3, that I want to continue studying in my future lab. So how I'm going to finish or to continue this aim, so I want to do some target metabolomics in Cryptosporidium, also to study the glucose flux in infected cells, do some spatial cartography, and complement with gene expression analysis. So with that, I just want to say thank you to my 2 mentors, Dr. Melissa Scala and Laura Knoll, and my collaborators in the SPEED project, and all my collaborators in my K99. To the leading edge, thank you so much. And here, pictures from my 2 labs, the Knoll lab, the parasitologist lab, and the Scala lab, the engineering lab. Thank you so much for your attention. Let me know if you have some questions.
I'm
sorry, I could be better at catching. So I was curious to know a little bit more about STAG and how treatment with that changes the gene expression during infection, and like what that indicates about those genes that you're going to study in the future.
Okay, thank you. So STAC has been studied before for other infections. I think was tested before for malaria, Plasmodium bergei, and reduced infection was tested again influenza, and also bacterial infection, I think was Listeria. And in all cases, reduced infection, but they never run gene expression analysis. So we infect the mice, we increase Cryptosporidium, we collect the fecal samples, and we found and we treat with STAC, and we found reduction on the shedding of oocysts. And then we do the gene expression analysis, and first STAC stimulate interferon type 2 response. So I think this is the main mechanism why it's reducing the burden of the parasite. And second, also, we found that downregulates this gene, lactase and glucose 6-phosphate and the fructose bisphosphate. So at this point, I don't know the answer, but I'm very curious to continue this project. But I don't know how STAC is downregulated this gene. We don't know. But I think it's very important, especially because lactase, so there is some preliminary data that say that people who is intolerant to, so some people become intolerant to lactase after Cryptosporidium infection. There is some preliminary data. But I want to follow up on this preliminary data and continue with that, but I don't have the complete answer yet. Thank you, Teresa.
So I had a question. I thought it was really interesting how your infected cells, they became, they had an increase in oxidation. And so I guess my question is two parts. One, is this something that typically happens to cells post-infection of any parasite? Is it specific to your parasite? And the cell becoming elevation and oxidation, is that actually help the parasite, or is that actually create a condition that potentially prevents it or, from becoming more virulent?
I think it's like somebody here was presenting it's like a battle. It's like a fight between the host and the parasite. So both have a antioxidant response. But at this point, I think we don't know what is the cause of that. We know that during infection, there is a regulation in the parasite of a lot of genes or transcripts related to redox biology, but also the host response. So it's like a balance between both conditions. And yes, the other question that if it's common in other parasites, yes. Using similar technique like FLIN of optical metabolic imaging, I have found that other bacterial infection, nobody has studied in parasite before, but other bacterial infections show also more oxidized environment over time.
Interesting. And then I guess it is a related question. I was curious, it seemed like you had a pretty global decrease in metabolism in your mice.
Mm-hmm.
I guess I just thought that was kind of interesting as to how it is so global. Does that make sense?
Yes.
You could speak to it?
Yes, in the Cryptosporidium infection, how these mice were infected and also treated with STAC and then regulate many genes.
Yeah.
Yes, I have never thought about it. I don't know.
It just seemed a lot wider than I don't know.
Yes.
Mm-hmm. But I'm also not that familiar with the field. So.
Yes, and also it's interesting because STAC came from Toxoplasma. So we are using Toxoplasma to treat Cryptosporidiosis. Mm-hmm. So yes, but this is what I want to continue studying. Mm-hmm. I don't know.
No, it seems really interesting. It seems very, very interesting. Any questions? Yeah.
Oh. Hi, that was a really nice talk. I have a question. I'm not sure. It's very much not my field. But how does your host cell deal with overproduction of various nucleotides without the infection subsequently?
So
is there any regulatory effect?
So how the host cell regulate the nucleotidases is the question?
Yeah, because you triggered this, but then you didn't let the infection happen. And so are you also seeing anything that the host cell is just
yes, that's a very interesting question. So far, we know that the single kiss and spit that doesn't have full infection, so the parasite cannot go inside, change many things in the whole cell. So we believe is these proteins that I present at the beginning, like the rhabtrias, the micronase, and the ferrical bodies proteins that are secreting during this kiss and spit, injected inside the host cell, and these proteins are the ones who are changing the metabolism of the host cell. It's not a full infection. And also because we try by optical metabolic imaging the kiss and spit, and we detect early changes, even at 1 hour or 15 minutes post-infection, we detect changes not only at the redox ratio, but also fluctuation in ADPH and FAD. So we believe it's all these proteins that are secreted during this kiss, or the combination of all of them. So my future project is to start checking to knockouts of these rhabtrias, to determine what is the most important of all these rhabtria proteins or spherical body proteins. But yes, so far we can say that a single kiss of the parasite can manipulate the metabolism of the host cell.
Can you prime the host cell so that if you do a conditional mutation, and then so the parasite can infect immediately, but then the cell has time to deal with what's happening, and then you wait half an hour or an hour, and then let the parasite infect? What do you think would happen?
I think we did some kind of similar with just the media. Just we have an infection, we take just the media without any parasite and add to the host cell. So this media doesn't have any kiss and spit, no parasites, and didn't change the host cell, didn't affect the metabolism of the host cell. So for that reason, we believe actually it's the kiss and spit that is changing the metabolism of the host cell. But I want to follow up with knockouts to see specifically what is that is changing. Did I answer your question?
Yeah. So much.
Yeah.
Okay, thank you.
Oh, sorry. Oh, where should we? So yeah, thank you so much.
Thank you.